Good morning, students. Good morning. So today 12th standard biology chapter 6 molecular basis of inheritance. In that particular chapters, properties of genetic material. Normally we know the genetic material is genes or DNA present inside the chromosomes or in the chromatin reticulum in the nucleus. In 1950s and before 1950s, the very elaborate search for the genetic material. Various scientists searching the genetic material where the genetic materials are present. So some scientists said inside the cell, some scientists said inside the nucleus, some scientists said inside the chromosomes, some scientists said inside the DNA, inside the gene or inside the cistron. Various topics are covered. But after 1950s only, after Watson and Crick, James Watson and Francis Crick, only the DNA is the genetic material. So various scientists explaining various concepts, various theories, various experiments. Some scientists like Hershey and Chase using a bacteriophage and bacterium for this research. After this research only, he proved that the DNA is a genetic material, is not the protein. And due to transforming principles, Griffith experiment, the scientists narrowly related to DNA, but not exactly identify the DNA. Something we transformed, but what is transformed? The scientists don't know the answer. So don't know the answers. So for various scientists, various research, after various experiment only, scientists concluded that the genetic material is nothing but DNA. RNA also acts as a genetic material in some viruses. Protein may be genetic material in those days, and DNA also genetic material. Normally for the competition for the genetic material, DNA is the one competitor, RNA is another one competitor, and the protein also another one competitor. The three competitors are available for the which one is better genetic material. So scientists provide some criteria for selecting this contest, selecting the particular molecule. So which one is a better genetic material? To following these criteria, some criteria are present. Following these criteria, we choose, we identified which one is a better genetic material. Okay. The properties of genetic material first talks. Normally we know the various scientists and various researchers is used a variety of genetic materials, like DNA as actual genetic material, and RNA act as a genetic material, even protein also act as a genetic material in some scientist words. So after final conclusion, we use some criteria for identifying the genetic material. These the genetic material fulfill these criteria, means the genetic material should be a better, best genetic material. Suppose only few of the criteria are fulfilled by the genetic material, means it is not a better genetic material. It is actually act as a genetic material in some organisms, but in universally is not accepted as a genetic material. Okay. Then we know that RNA also act as a genetic material, ribonucleic acid. Normally we know that nucleic acid is two types. One is deoxyribonucleic acid and the ribonucleic acid. DNA means deoxyribonucleic acid are present inside the DNA of eukaryote as well as prokaryote organism. Even some viruses, RNA also act as a genetic material. For example, TME viruses. TME viruses means tobacco mosaic virus. We know the structure of TME virus, which causes tobacco mosaic disease in tobacco plant, and is actually an RNA virus. It is the virus genetic material in RNA, not DNA, because we know the virus, any one, maybe viruses contain DNA or RNA, any one of the genetic material only present. The next one is QB bacteriophage. Bacteriophage means what? The virus which kills the bacteria. The T4 bacteriophage, the lambda bacteriophage, QB bacteriophage, T4, T7, T odd and T even pages are present. So bacteriophage is nothing but the virus which kills bacteria and cause disease in bacterium, that is bacteriophage. Even some bacteriophage and TME also is contain genetic material as actually RNA, is not a DNA. So what are the criteria for selecting the genetic material? So it should be able to generate its replica. Replica means replication. So those substance which replication, it performs replication naturally without the involvement of any other organisms. One genetic material, not a genetic material, but a biomolecule which which performs replication, means that a particular is known as genetic material. It is the best genetic material. Normally, you know the three competitors are for the genetic material, that is DNA, RNA and protein. DNA also replicate. We know that DNA performs replication in S phase of the cell cycle. Even RNA also replicate. In some viruses, RNA also replicates with the use of some substances, some enzymes like reverse transcriptase and polymerase and various with the use of various enzymes. RNA also replicate, but protein is not replicate. We give one protein, for example histone, a protein present in DNA. Then histone protein is not directly is replicated into another histone. No. Our muscle cells contain various protein like actin, myosin, meromyosin, troponin. Various proteins are present. Even actin is not individually, it is not able to replicate. Even egg contain albumin, globulin, milk contains casein, a variety of hairs and nails contain keratin. So various protein are available in the environment or in the food materials, but the protein is not has capacity to replicate. DNA has capacity to replicate, RNA also has capacity to replicate. In the first point itself, the protein is not fulfilling the criteria. So for the genetic material should follow all these criteria. The first, protein is not fulfilling the criteria, first one. So protein is not act as a genetic material. Only DNA and RNA act as a genetic material from the first point. Then the second point, it should stable chemically and structurally. It should be stable. Stable means it is not changes over the temperature, over the heat. So it's not change over the heat and temperature as well as any other environmental problems. From the first point itself, the protein is not a genetic material, because it is not replicate, it is not has capacity to replicate. Then second point, it should stable and chemically and structurally. So normally the DNA is stable because due to our experiment like transforming principle, Griffith experiment, estrine after heating are mixed with austine. We know estrine bacterium, that is Streptococcus pneumonia bacterium with estrine, after heating it is mixed with normal austine bacterium. Then after injected into the mice, the austine turn into estrine. So due to heat, the DNA of the estrine is not damaged. Some of the properties, not fully not damaged, some of the property of DNA is not damaged due to heat in the particular experiment, that is transforming principle experiment. So with the use of these experiments, we conclude the DNA is actually chemically and structurally stable as compared to the RNA. Now how the chemically structurally stable means, normally in each DNA is contained thymine instead of uracil. So in RNA, uracil are present. Suppose in DNA means adenine, thymine, cytosine, guanine. 4 nitrogen bases are present. Already I have studied. 4 nitrogen bases are present. Like we know that is DNA means adenine, thymine, cytosine and guanine. And nitrogen bases are present. The thymine is actually a stable, basically structurally, chemically stable nucleotide. Basically it is a strong nucleotide. It is not degraded with the use of like uracil. In RNA means adenine, uracil, cytosine, guanine are present. But in uracil in the RNA is structurally and chemically not stable. In DNA, the adenine are present. Then adenine and thymine is structurally stable, and uracil instead of thymine, in RNA uracil are present. Uracil is structurally not stable, chemically also not stable, because each DNA contain only ODAS hydroxyl group in last end of the particular DNA. But in RNA, every nucleotide OH group are present. So hydroxyl group are present in every nucleotide of RNA, but DNA only one hydroxyl group are present. So in the end, so the hydroxyl group also it is present in the RNA, it is become weaker due to the presence of uracil and due to the presence of hydroxyl OH group in every nucleotide. That the particular RNA actually is structurally unstable, but due to the presence of thymine and due to the presence of hydroxyl group in only one in the DNA strand, it is actually stable. So DNA become more stable chemically and structurally. We proved with the use of the stableness and the particular structuralness in the transforming principle as well as this one. So due to the presence of thymine, the DNA become more stable. Due to the presence of uracil and every nucleotide has 2'OH group, due to the presence of these two factors, RNA become unstable. So the second criteria for the better genetic material is it should be stable and chemically and structurally. DNA only stable and chemically, structurally. So DNA is a better genetic material. RNA is unstable. So the RNA is not act as a genetic material. Maybe in some viruses RNA act as a genetic material, but majority of the organisms DNA only the genetic material. Next one, scope for slow changes that useful for the evolution. So scope for slow changes means the organisms, like example human beings. Human beings are developed from where? That is monkey. Homo erectus, Homo habilis, slowly chromocron and all the organisms slowly changes into human beings. Human evolution is approximately, it takes 2 million years, not 100 or not thousands of years. 2 million years. After 2 million years only, monkey like apes slowly changes into modern human like Homo sapiens. Okay, so its slope for the DNA, due to the presence of DNA only, it slowly changes. But RNA is mutate faster rate as compared to the DNA. For example, the normal disease in our society nowadays, COVID-19. Corona actually as the 19 is RNA virus, and HIV also RNA virus, human immunodeficiency virus, and Ebola, SARS, severe acute respiratory syndrome, and swine flu, H1N1 virus, Pandrikachal, we are called it as Tamil, H1N1, hemagglutinin 1 and neuraminidase 1. These type of viruses are RNA virus. It is actually structurally not stable. It is mutate faster rate. Normally the genetic material means slow mutation occurs. Slow mutation, slow changes in the DNA, it is important for the evolution. But in RNA means mutate faster as compared to the DNA. It is easily formed into another organism. So it's controlling the particular pathogen, of controlling the particular disease is slow. It takes long time to control the particular disease. Okay. That the third point, scope for the slow changes, that is useful for evolution, but DNA is useful because it's scope for the slow changes, not a faster mutation like RNA. But RNA is not forming a slow changes, it is faster rate, mutate faster rate, so it changes into another form of RNA in a short period of time. So DNA is a better genetic material as compared to the RNA from this point also. Then it should be able to express itself in the form of Mendelian characters. So Charles Mendel, John Mendel, it follows various theories like principles of inheritance and law of dominance, law of segregation, law of independent assortment. These are three Mendelian characters like alleles and the genes. All these characters is followed by the genetic material. But RNA also follows some but the Mendelian character. DNA also some follows the Mendelian characters. So according to this criteria, majority of the living organisms contain DNA, act as a better genetic material. RNA also act as a genetic material, but in some organisms. Okay, according to these criteria, protein is not a genetic material because protein it's not follows these criteria. Okay, so criteria for selecting the genetic material means RNA is a better genetic material in majority of the organisms. DNA is not RNA, sorry, DNA is a better genetic material for the majority of the organism. RNA also but genetic material, but in some viruses. Okay, yes. So which one is a better genetic material according to this last criteria is DNA is a better genetic material. Okay, so DNA is a better genetic material, we know, but according to this point of view, which genetic material is evolved faster or evolved earlier? So RNA is evolved earlier. So earlier means RNA become the first genetic material millions of years. For example, human. Normally, according to the Big Bang theory, Earth evolution, Earth formation is around 4,500 million years. So, 4,500 million years before only Earth is slowly developed. But after that, the living organisms like unicellular organisms developed in around 3,000 million years. So, first evolved organism, first evolved genetic material maybe RNA. Then after some modifications, because we know the RNA is not stable, it is unstable. So due to some modifications in the RNA, so RNA slowly evolved into DNA. So now we know the DNA is a better genetic material in all other organisms except a few viruses. So earlierly first formed maybe genetic material, maybe RNA, and slow some structural modifications in the RNA slowly evolved into DNA. So but some reasons, because all the metabolism, for example, our cell producing one protein or enzyme, for example, in salivary gland. In our body, a mouth contains salivary glands like parotid, submaxillary and sublingual salivary glands are present. The salivary glands are producing maybe enzymes like ptyalin or maybe salivary amylase. The salivary amylase is an enzyme, this is actually a proteinase enzyme produced from the ribosomes of salivary gland cells. So ribosomes need mRNA, tRNA, rRNA for the production of enzymes. So RNA is important for the production of enzymes. So all this metabolic work, metabolism means what? All chemical reactions are performed in a living cell. A living cell performs various chemical reactions like respiration, photosynthesis in plants, metabolism are various chemical reactions like protein synthesis, hormonal synthesis, interaction between one cell to another cell, like a movement of substances like active transport, passive transport. Various chemical reactions are occurred inside the cell. So, some various chemical reactions are collectively known as metabolism. Some chemical reactions are formed newly, or maybe material are formed newly. For example, during photosynthesis, glucose is produced newly. So, that is actually anabolism. During respiration, the glucose is breaks and forms energy. Glucose is breaking. So, the process is catabolism. So, anabolism and catabolism is collectively known as metabolism. So, metabolism is nothing but all chemical reactions performed in a cell, in a living cell is known as metabolism. So, metabolism are around in RNA only. So, all the type of metabolic reactions they need enzymes. Enzymes means it's actually a proteinaceous enzymes. Protein means with the use of mRNA, tRNA and rRNA only the proteins are produced. So metabolism need RNA. And translation means the mRNA into protein. In next section what is meant by translation, transcription I explain. Translation is nothing but formation of protein from the RNA. So with the use of RNA only the protein are produced. So, the RNA is important for the translation process. Without RNA, translation process is not taking place. And splicing we know in the next section, the removal of introns from the cistron. So, normally in H and RNA are produced. Actually, in the next section, transcription, translation, the splicing are present. The removal of introns and formation of matured mRNA, that is splicing and evolution of around the DNA. Evolution means all these process, metabolism takes place with the use of RNA. Translation also takes place with the use of RNA and splicing are placed in the RNA. So, all some main functions in the cell around RNA only taking place. So, RNA is important for expressing the character, for producing the enzyme, for producing the character, phenotypic expression. For example, one person become tall. How the person become tall? Due to the production of growth hormone from the pituitary. How pituitary produce growth hormone? GH. So, pituitary has some cells. Pituitary has some cells, has some genes, DNA. The DNA has message for the production of growth hormone. The DNA giving message to the mRNA. mRNA get the message from the DNA and the message is given to ribosomes with the use of rRNA and tRNA. The growth hormone is produced because hormones are produced inside the cell in the ribosomes. Ribosomes only producing the particular hormones. So, ribosomes producing hormones actually this is actually proteinaceous hormone. So, hormone is actually a protein, some hormones, some serial hormones are present, hormones normally protein. So, the protein production, the hormone production which is important for the growth. So, more hormones are more growth, more tall. So, here the expression of tallness is controlled by hormone. The hormone production which controlled by RNA, and RNA also is controlled by DNA, but without RNA our growth hormone is not produced. So, RNA is important for our metabolism because growth is one type of metabolic reaction. So, the growth is occurs due to RNA. So, all these metabolic reactions and translation, all this process with the use of RNA. Without RNA, our physical and biochemical process like metabolic process not taking place in the cells, in the living cells, even plant cells also, in animal cell also. Then, RNA is catalyst and unstable. Catalyst means it's enzyme, we know the biocatalyst enzyme. Give an example of biocatalyst. Salivary amylase is a biocatalyst. Tylin, pepsin, renin, carboxypeptidase, mucopeptidase, nuclease, DNase, RNase, nucleosidase, nucleotidase. sucrase, lactase, maltase, various enzymes present in our digestive system. So the enzymes are produced from the exocrine gland. So these enzymes is not a enzyme, but it's not as a RNA, but some RNA act as a catalyst in some process, in replication and in the transcription process, and the next class replication and transcription. In RNA also act as a catalyst, which is important for the stimulating the reaction. What is meant by catalyst? The substance, the chemical substance which is stimulate the reaction, which is induce the reaction, it induce the metabolic reactions in the living cell. That particular substance is actually catalyst. So here biocatalyst means it increases the biological process, biological metabolism, chemical reactions inside the cell. So RNA also act as a catalyst. So normally we know catalyst enzymes means due to pH, due to temperature. In 11th standard we have studied, due to pH and temperature enzymes are modified, because I give an example. Salivary amylase present in our body, which act as a 6.9 pH and temperature around 37 degrees Celsius. Only it acts on starch and converts the starch into maltose. But due to increasing temperature, due to fever time, our salivary amylase is not performs properly digest the starch, not properly digest the starch, because due to increasing in temperature, increasing in pH, or maybe decreasing in temperature, decreasing in pH, enzymes is not performed properly. So RNA also act as a catalyst. Particular time it's due to temperature it is modified, so it is unstable. Stable means is not stable means due to chemical, due to temperature, due to pH change, it's become stable. Then only it's known as stability. RNA becomes stable due to Griffith experiment, transforming principle experiment. We conclude DNA is a stable. Due to temperature also it becomes stable. But RNA acts as a catalyst. It's actually unstable. Due to temperature it is changed their shape. Automatically the characters also changed. So majority of the viruses like RNA viruses become unstable due to the RNA only. Okay, these are all the why RNA is the first genetic material, then how the RNAs become slowly evolved into DNA. So which one is the genetic material, best genetic material? That is DNA. So DNA is the better genetic material from the above decision. So what is mean by replication? So next DNA and RNA also over. The next one is replication. What is mean by replication? One DNA is converted into two DNA. Normally one DNA helix are present, that is double helical structure of DNA, and double helical structure of DNA slowly separate and forms another one DNA helix, mean that is replication. The replication is the replication procedure is actually postulated by the Watson and Crick in 1953. Normally DNA, the semi-conservative plus and particular structure of DNA is identified and discovered by the Watson and Crick, James Watson and Francis Crick, and in 1953 he immediately postulated some explaining some procedures regarding replication. How this original quotes, original message from the Watson and Crick, it has not escaped our notice that the specific pairing of we have postulated immediately suggests a possible copying mechanism for the genetic material. It has not escaped our notice. It has not escaped our notice that the specific pairing, this is correct procedure, immediately we suggest the procedure for the DNA replication. So generally is not escaped from our notice, the particular Watson and Crick. So we immediately suggesting the copying mechanism of a genetic material like DNA. Okay, what is material of what is the replication and how the replication is experimentally proved with a semi-conservative method of replication in the next section. So first I give basic idea about what is meant by replication. What is meant by replication? The one DNA is copied into another DNA. The process is replication. Again, the Watson and Crick, the original quotation, it has not escaped our notice that the specific pairing we have postulated, the pairing of replication we have postulated immediately. We postulated immediately means the Watson and Crick postulated immediately, suggesting a possible copying mechanism. Copying mechanism, that is replication procedure. How the DNA is replicated. The procedure or the replication experiment is it's immediately suggested by the Watson and Crick. What is meant by replication in the next? So normally we know. So replication means what? So normally we know the DNA. Then one strand of DNA. Next to this is another strand of DNA. Okay. The one strand normally DNA contain 5' to 3'. Means another strand contain 3' to 5'. 5' means 5 prime. That is actually DNA. During replication procedure, first DNA is uncoiled. Uncoiled means listen. This is actually DNA, or this is 5'. And this one 3'. So first, during replication procedure, first DNA is uncoiled. Uncoiled means separate. Two strands are separated. So first, during replication procedure, the DNA become uncoiled. The first, normally the DNA is a double helical structure. The double helical structure, there is a hydrogen bond represent. The hydrogen bond slowly, this is due to the isomerase, due to topoisomerase, and due to helicase. And what is meant by replication, I give explain in the next section. I give a small idea about replication. So DNA is normally is a double helical structure according to the Watson and Crick view. So DNA is replicate during cell cycle, during S phase of the cell cycle. Okay. Then the DNA is actually, this is 5' and the 3' and 5' and the 3' is slowly is uncoiling, is separating. Now this is 5'. Here that is 3'. Because due to the replication procedure, DNA slowly separates. DNA strand, double helical structure is separate. Then with the use of DNA polymerase, DNA dependent DNA polymerase is actually an enzyme. DNA dependent DNA polymerase is an enzyme. With the use of DNA polymerase, new DNA are slowly formed. For example, here adenine means here thymine, cytosine means guanine, guanine means cytosine, cytosine means guanine. Slowly the DNA formed. Supposing your 5', for example, if 5' means actually this is not an exact method. It is actually 5' to normally DNA replicate towards 5' to 3'. Okay, here 3'. Here 3' means you change. It is 5'. 5' means here 3' and 3' means opposite side 5 dash. This is the correct sequence. So DNA is actually one strand starts from 3 dash to 5 dash, another strand starts from 3 dash to 5 dash. So after uncoiling, one strand is actually a parent strand. Here is actually a parent strand. So here also a parent strand and here also a parent, because parent strand only divide into two strands. Then new strands are formed according to the parent strand. For example, in parent strand, the parent strand actually a template strand. Adenine means the daughter, the newly formed DNA is thymine, cytosine means guanine, guanine cytosine, cytosine guanine. So normally DNA it replicates only in 5 dash to 3 dash direction. DNA replicates only in 5 dash to 3 dash direction. This is a rule, common rule for DNA replication. Even RNA also. I give an elaborate idea in the next section. That is by replication and what are all the enzymes are involved in the replication. All this in the next section. Okay, I give a small idea about replication, right? And the next section I give elaborate and full description about the replication. So due to this 5 dash to 3 dash a new RNA are formed. And afterwards here from this side DNA also formed in 5 dash to 3 dash. So in replication procedure, one strand acts as a parent strand. This is parent, because parent strand only is divided into two. This is actually a parent strand, and this one also actually a parent strand. The parent strand is known as template strand. Template strand means it is useful for the synthesizing a new DNA. That is actually a parent strand. Here also a parent strand. And this is actually a newly synthesized strand. Newly synthesized strand means newly formed strand. And here also a parent strand. Here this is newly synthesized strand. This is actually discontinuous strand. Here this is 5 dash to 3 dash is continuous strand. Here is forming discontinuously. So this is discontinuous strand. So this strand is actually a newly formed strand, and this strand also actually newly formed. So then both the strands, one strand is old strand, one strand is a new strand, and both the strand is joining. And form a new DNA. Here also, with the use of DNA ligase, the strands are pasted. Then I give another idea. Next section. So totally one DNA, two helical structure DNA. After replication procedure, two replication in one replication. 2 strands of DNA, 2 DNA are formed. This is actually replication. Replication is nothing but once DNA helix is separated with the use of another strand and some enzymes, the DNA become 2 stranded, 2 DNA. This is actually daughter DNA is also daughter DNA. One newly synthesized DNA, as is daughter DNA, contain one parent strand. And in the yellow is newly synthesized strand. So all the DNA contain one parent strand, another one newly synthesized strand. Now parent strand is conserving in our DNA, our cell DNA also. One cell DNA contain one DNA strand is actually parent strand. Parent means before cell, and newly synthesized DNA 1%. Though type of replication is semi-conservative method of replication, because it's conserving the one parent strand, and one parent strand is newly synthesized. It conserves the one parent strand and one newly synthesized strand are produced. So this phenomenon is known as semi-conservative method of DNA replication. I give an experimental proof also in the next class. I give a small idea about replication. Replication is nothing but one DNA is converted into 2 daughter DNA. The process is replication. Explained by Watson and Crick I give a small idea about replication. In the next section, what is meant by replication and what are all the experimental proof of replication. Okay.